The mammalian tongue contains numerous taste buds. Each taste bud has a taste pore. Through the taste pore, fluid carries solutes inside. The taste pore consists of microvilli. Each taste bud consists of receptor cells. The number of receptor cells or taste cells in a taste bud is about 50 to 100. The taste bud cells are continuously replaced by cell divisions. The receptor cell or taste cell is a type of epithelial cell. Taste cell shows some of the function of neurons. Each taste bud is surrounded by epithelial cells. Nerve cells are supplied to the taste cells. These nerve cells forms gustatory nerve fibers which go to brain. In order to detect the salt, the plasma membrane of receptor cell or taste cell has amiloride sensitive Na plus channels. The amiloride sensitive Na plus channel is otherwise called as ENaC channel. The extracellular Na plus ions move through the ENaC channel and reach to the cytosol of receptor cell or taste cell. The inward movement of Na plus ion is called as influx of Na plus ion. Due to this influx of Na plus ion, the taste cell depolarizes. ENaC channels detect salt in insects like Drosophila. Other members of ENaC channel family play important role in neural memory. In order to detect acid or sour taste, the receptor cell or taste cell's plasma membrane has amiloride sensitive Na plus channels. The H plus ions present exterior to the taste cell moves through the amiloride sensitive Na plus channel and reach to the cytosol. Due to this influx of H plus ions, the taste cell depolarizes. In another way, to detect the acid or sour taste, the taste cell employs K plus channels. The K plus channel is present in the plasma membrane of taste cell. The H plus ions present exterior to the taste cell move through the K plus channel and block it. As a result, the cytosolic K plus ion movement through K plus channel is blocked. As a result, intracellular K plus ion or positive charge increases. As a result, a depolarization is created. In order to test amino acids, the receptor cell or taste cell depends on G protein coupled receptor or GPCR. The GPCR is coupled with G proteins like alpha subunit, beta subunit and gamma subunit. The monosodium glutamate is one of the most widely used food additives in commercial foods. Monosodium glutamate gives a special aroma to processed foods. This special aroma is known as umami in Japanese. The monosodium glutamate binds to the GPCR and gets sensed by the taste cell. Umami tastes are detected by protein family related to the T2RS called T1RS. T1RS form homodimers and heterodimers. In order to test bitter compounds, the taste cell uses divalent salt or quinine sensitive K plus channel. The bitter stimulus, for example quinine, present exterior to the taste cell moves through the divalent salt or quinine sensitive K plus channel and blocks it. As a result, the cytosolic K+ ion movement through divalent salt or quinine-sensitive K+ channel to the exterior of the test cell is blocked. As a result, the intracellular K+ ion or positive charge increases. This increase in positive charge produces a depolarization. Bitter molecules can be detected at concentrations as low as 10 to the power minus 12 molar. In another way to detect bitter compounds, the test cell uses G protein coupled receptor or GPCR. This GPCR is coupled with G proteins. The G protein has 3 subunits like alpha subunit, beta subunit and gamma subunit. Besides these proteins, the test cell also uses the phospholipase C protein to test bitter compounds. During the detection process, first of all, the bitter stimulus present exterior to the test cell binds to the GPCR. The G protein coupled to the GPCR then activates phospholipase C. The phospholipase C then catalyzes a biochemical reaction in which IP3 and other products are produced. Due to this IP3 production, the cytosolic concentration of IP3 increases. Due to this increased IP3 concentration, the Ca2+ ions are released from the intracellular compartments like the endoplasmic reticulum. As a result, a depolarization is created. Now, some important points. In human, an important bitterness detection gene is present on chromosome 5. One G protein involved in bitter test transduction is called as gosdeucin. Bitter tests depend on about 25 genes encoding various T2RS. T2RS are test receptor protein with 7 transmembrane alpha helical domains. T2RS are members of the class of proteins known as GPCRS. About 15% of all test cells express T2RS. In another way to detect bitter compounds, the test cell uses a different strategy. In this case, the G protein coupled receptor or GPCR is coupled with G protein having alpha subunit, beta subunit and gamma subunit. The bitter stimulus present in the exterior of test cell first binds to the GPCR. The G protein coupled to GPCR then activates the cytosolic PDE or phosphodiesterase. The CAMP or cyclic AMP then binds with the active PDE. In a PDE catalyzed biochemical reaction, the bound CAMP is converted into AMP or adenosine monophosphate. The AMP is then released into the cytosol. Like this, the cytosolic cGMP or cyclic guanosine monophosphate binds to the active PDE. In a PDE catalyzed biochemical reaction, the bound cGMP is converted into GMP. The GMP is then released into the cytosol. Due to such biochemical reaction, the cytosolic CAMP concentration decreases. This decreased CAMP concentration may cause the closed ion channels to be opened. Through opened ion channels, the Ca2+ ions present exterior to the test cell move into the cytosol. As a result, a membrane depolarization occurs. Please like, subscribe and share.